this is where we left off, right? Talking about the aldoses and ketoses. Okay, so any questions about what we covered so far? I'll take that as a no. So, remember we classify sugars based on the number of carbons that they have. So we have like 3 carbons, that would be a triose, 5 carbons, pentose, 6 carbons, a hexose. And we can also classify them by the position of the carbonyl group. So remember that functional group you guys are supposed to learn from chapter 4. So what's a carbonyl group? Or maybe nobody learned it. Just a reminder, okay, so a week from Thursday you have an exam on this stuff. So a carbonyl group is C with a double bond, so it can be like that to an oxygen in the middle of a carbon chain, or it could be at the end of a carbon chain. So this one is called a ketone, and so in a sugar it's called a, and this one is called an aldehyde, and so in a sugar it's called, so that's how we can name and classify sugars. So on the test I might draw this and tell you that's a sugar. What is that? And you'll have to pick it out of a multiple choice lineup. So is it a triose, a pentose, a hexose, an octose? None of the above. Yep. No, three carbons, right? Yeah. So it's usually 1 to 2 to 1. But 3 carbons, it's a triose. 5 carbons, like this one, 1, 2, 3, 4, 5. What is that? It's a pentose, right? I might put this on the test and ask you, is that sugar aldose or a ketose? Oh yeah, it's in the list. It's aldose. All these are aldoses, right? These have the same formulas, right? So what do we call something with the same formula but a different shape? Isomer, good. So this is definitely an isomer, right? Because this is a ketone bond here, as opposed to glucose, which is a aldehyde bond because it's on the end. So these are different shapes. But what about these two? Are those isomers? So you wouldn't think so, because in this form they're free to rotate. So I could spin this group with the OH over here, and the H would go over there, right? And that would look just like this one. But the problem is that sugars don't exist in this state normally. What they exist in is rings, and that changes things. So this is the normal state of glucose, which is in a ring form. And if we talked about single sugars, those are called monosaccharides. We put 2 sugars together, that's a disaccharide. And then if we put more than, we call it a polysaccharide. So more than 2 is a poly. Any questions about that? Yep. Right, so it has a ketone bond in it, right? So if you're not sure, you should go back and review the chapter 4 slides and make sure that you know the difference between ketones and aldoses or aldehydes in the carbonyl group. You need to know all 6 of those functional groups we talked about. You need to be able to identify those on the test. All right, so here's glucose in its ring structure. So this is it linearly, but this can form a ring, and this is the ring structure. So you can see that here we have a H and an OH, and the reaction to link 2 glucose molecules together is called what? dehydration. So we take out water and we form what's called a glycosidic linkage, which you can see here. So this, I drew the structure of DNA before, and remember we numbered them, and I showed you how to number all the stuff in these groups so we can identify where everything is. So sugars are no different. In a ring structure, we number them clockwise, starting with the oxygen. So this is number 1, 2, 3, 4, 5, and 6. So the numbers are put on here. This is showing you that this is a number 1. We see that? Because we start here at the oxygen, and we number 1, 2, 3, 4, 5, 6. Again, this is another glucose, so what number is that one? And this one? And so you know you're right, because they put a 4 there. So this is a bond, it's called a 1,4 glycosidic linkage, because it's between the number 1 carbon and the number 4 carbon. Now, why does that matter? It matters because it changes the shape of the molecules. And remember, in biology, the most important thing is shape. So, you know, there's a difference between sugars that you can eat and digest and sugars that you can't. Like cellulose, what does that end in? So what does that tell you? So that's what makes up tree bark. Can you go eat tree bark and live off of that? But you can't live off of it, right? You can't digest that. You can't digest cellulose. So the difference between cellulose and carbohydrates is very small, right? It's just a bond. But that is huge to your body. That's what we've been talking about this whole time. So that's why you need to know this. So on the test, I'll ask you what are the kinds of bonds between these different sugars, and you're going to need to know them. So I'll give you examples as we go along. So we link 2 glucoses together, and that makes a maltose. That's the name of that disaccharide. So 2 glucoses linked together is maltose. Multiple glucoses linked together are, you guys know? It's called carbohydrates. But that doesn't end in OSE, so the scientific term for that is, remember, amylose. That's right. Alright. So just to give you an example, we have different kinds of sugar. So this is maltose. We have glucose linked to fructose. That's another disaccharide, right? But that makes a different kind of sugar. That's sucrose. You guys know what that is? Well, it's regular sugar you buy in the store, right? You go buy a bag, it says sugar on it. That's sucrose. Or table sugar. But this one has a different linkage, and the reason that this is numbered weird is because this molecule is flipped around. So if you turned it the other way, it would be numbered 1, 1, 2, and so on like that, but this one's rotated. I don't care that you know these rules. I'm just showing you this so you know that there's different kinds of bonds. So all you need to know is the bonds. I'm going to ask you what kind of bond is in a sugar, and you need to be able to tell me 1, 4, 1, 2. And the reason I'm showing you this is so you know that's just not some silly numbering system. So you go clockwise. There's some chiral rules and stuff like that. Don't worry about it. All right? I don't want to get into IUPAC numbering of organic chemicals in this class. Just know that there are numbers for each of the carbons. Okay? Don't worry about the rules. You'll learn that in organic chemistry. Alright, so polysaccharides are made up of monomers, right? And they're used for different things. So animals, plants, different species use them as storage of energy. So plants sit out in the sun all day. Their job is to take in carbon dioxide, which is what they're breathing. They combine that with water, and they rearrange molecules to make glucose. So that's what they're building. That's the purpose of photosynthesis, is to make glucose. And then what do when they get a bunch of glucoses made? They link them together and they make what, right, which is you call carbohydrates, and they store them. Because when the sun goes down, they can't make sugars anymore. What are they going to do for energy? Burn them, right? Just like you would. So they digest them, the stored carbohydrates, and they use those for energy, just like you would, which you do, right? You sneak up on the plant and you steal its carbohydrates. It didn't make those carbohydrates for you, it made it for itself. But you're using its carbohydrates, right? Why are you doing that? Because you're mean? Because you can't do photosynthesis. So you have to rely on plants for that. And all organisms do. Alright, so plants store their energy as starch, which is the same name as a carbohydrate, which is the same name. The correct name, the scientific name, is amylose. That's the name of the sugar because it ends in OSE. But they all mean the same thing. Yeah. So those are all the same names. And all it means is there's lots of glucoses linked together in long chains, like beads on a string, right? It's easier to store things that way than just to have them all over the place in your cells. Do we store our energy as starch? How do we store our energy? Fats, right? So, but fats take a little while to break down. So the reason that animals store energy as fat is because if I had a gram of fat, which is we're going to study in the next couple of slides, it's called triglyceride, and I had a gram of amylose, which you guys would call carbohydrates. This one has 4 calories. The carbs have 4 calories per gram. The fats have 9 calories per gram. So if you stored your energy as carbohydrates, pound for pound, you would need twice as much mass. And so would that do you any good if, let's say, you're out, hanging out in your backyard and all of a sudden a bear comes out? You got to run, but you weigh twice as much as you do now. Slow you down, get eaten. So it's not beneficial for animals to store their energy that way. Trees, they don't care. When's the last time you snuck up on a tree and said, "Boo, I'm going to get you"? What'd it do? Run away? No. So they don't care how they store their energy. It's the easiest way for them to do it because they don't have to move. But we can't readily access our stored fats. So we have to store our energy in a different form of carbohydrate called glycogen. So we don't store a lot of it because that would make us heavier, right? We only store about a day's supply of this energy. And the reason is like, let's say that, so you have fat stored in your adipose and your fat cells. So let's say the bear comes out and all of a sudden you're like, okay, hang on, I gotta release the fat from my fat cells, travel through my bloodstream to other cells so that they can use it to break down. Oh, you're eating. Right? That takes a long time. So glucose is an instant burst of energy, and so that's why your body stores that in case of an emergency. If you ever go to a gym or get on a piece of cardio equipment, you see that there's different levels of activity. Is the fat burning level the highest one? Why not? Wouldn't it make sense if you worked harder, you'd burn more fat? Why don't you do that? Why doesn't it work that way? Right. You burn through glycogen because your body thinks, oh my gosh, he's running away from a bear, or he or she is running away from a bear. I need that instant energy. I don't have time to go, let me just release some fat and be eaten. Does that make sense? So the fat burning is always lower intensity because you want to trick your body into thinking, he's just out, chopping wood or doing whatever. That way it knows that you're not in immediate danger, and it'll preferentially use your fat storages because it knows it has time to process that. Does that make sense? So glycogen is our short-term energy storage, and then organisms also use carbohydrates for structural support. So we're going to talk about cellulose, which If you look on your nutritional label, that is called fiber, right? And then chitin, which is the principal component that makes up like insect shells, like lobster shells and stuff like that. So these are all modified glucose molecules. I'll show you the difference between them now. So here's starch, right? Also known as carbohydrates or amylose. It's the polysaccharide that's found in plants. We don't store energy as starch, right? We store energy as fat, but we store short-term energy as glycogen, which is different. And I'll show you the difference. So this one, remember the numbering system, 1, 2, 3, 4, 5, 6. So this is a 1, and then 1, 2, 3, 4, 5, 6. So this is a 1, 4 linkage, glycosidic linkage, which I showed you before. But this one is called alpha. And the reason it's called alpha is because these sugars have a specific shape. So so here we have a carbon. Here's another carbon. This is our number 1, 2, 3, 4 carbon. If you notice on this one, so we have carbon. How many bonds does carbon have to form? So on this we have our ketone group, right? Because it's in the middle. So this is a double bond to an oxygen. Single bond to an oxygen, and then from that we have a hydrogen bound to it. So how many bonds does that carbon have now? Is that enough? So what do I have to put up here then? No? The capper. It's not drawn on there, but yeah. So we have the same thing over here. Alright, so we have these 4 bonds. Here's the question. Are those the same? The answer is no, they're not. Why? Yeah. If let's pretend that's an orange and these are sticks. If I shove sticks at an angle in an orange, can you spin it? If they're in straight, can you spin it? Yeah. But if they're at an angle, you can't spin it. Because this is in a ring structure, that is different than this. What do we call this one? I'm going to put something just like this on the test, and you're going to have to tell me, what is this called? First of all, what kind of isomer is this? It's caused by, it's geometric, that's exactly right. So when you have geometric isomers, there's 2 forms. What are they? No? So important that this is what we call an alpha glucose, and this is what we call a beta glucose. Because they're different. They have a different shape. Everyone agree to that? So this is called alpha bonds because it's made between what kind of glucose? Alpha, good. It's made from alpha glucose. So these are all alpha glucose molecules, and you guys built these in lab, remember? So when you if you built this, you would see that it would start forming a spiral shape. It's just the way the molecule is designed when you link it together. So this is long chains that cause this helix, and this, if it's straight chain, it's called amylose. If it's branched, it's called amylopectin, which is stuff they use to make like jelly and jams, pectin. And then in lab, we'll look at these. These are called amyloplasts because they're granules that store amylose, which is carbs or starch. They can't rotate. I can't rotate this to make it look like that. Because if I wanted to make this look like that, I would have to spin this bond. That makes sense? And I can't spin it because there's two sticks in the ring coming at different angles. Go home and get an orange and shove two sticks in it, and then try to spin it. Can't do it. If it were a straight bond, like a linear one, you could spin it, and they would be the same. But because it's in a ring, they're not. That makes sense? So those are geometric isomers, and I have a feeling that you guys might need to study that as well. Just guessing. All right. So amylopectin is branched. On the test, I'm just going to tell you, I'm going to ask you, what are the bonds that are found in amylose? What are they? What type of bond? Alpha 1,4. What's the difference between amylose and amylopectin? What's the bond in amylopectin? It's on the screen. You guys are getting nervous, huh? Because you haven't studied. Yeah. Yeah. So that question I just asked you is exactly what I'm going to ask on the test. I'm going to say, what bond is found in amylopectin? And you're going to have 5 choices, right? Because it's a multiple choice test. So it's going to say alpha 1,4 linear, alpha 1,4 branched, beta 1,2, alpha 1,7. None of the above. I love that one. No, because I throw that in a lot, so that makes it from a 5-choice multiple question to a lot larger one, right? Because it could be anything. So, what are you guys gonna pick?
Alpha 1,4 branch.
There you go, alpha 1,4 branch. All right. So, plants store starch in plastids, right? They're these little granules. They store their glucose in there, like we talked about, so that they can use it later on when they need it as energy. They also use it as a building structure. It's kind of their Tinker Toy set, because they're really their only carbon source, their only source for sugars and energy is to make them and pull it out of the air. And then you break them down, right? So they build up the glucose, And what's the reaction that plants have to do to build up glucose? That's another good test question, right? What's the reaction that plants do to make starch? It is photosynthesis, but I'm looking for the kind of reaction to build polymers, like starch. Dehydration, and then you put it in your mouth, and what reaction are you doing? Digesting it. Hydrolysis. Make sense? So make sure you guys know that. It's going to be on the test. So glycogen, we talked about what this is, right? We store this. Animals store this. You wouldn't find this in plants, right? And you wouldn't find starch stored in your body either. This one is unique. It's different than starch or amylose or carbohydrates, all the same thing, because this is branched alpha 1,4 and alpha 1,6, right? So that means it's linked between the 1 carbon and the 6 carbon. Does that make sense? So glycogen is alpha 1,4 and alpha 1,6. So on the test, I might ask you, what's the bonds in glycogen? And I'll have a list. Alpha 1,4 only, alpha 1,4 and 1,2, alpha 1,4 and 1,6, beta 1,4, gamma 1,7, just make it up. What's the answer? There you go. You got the question right. If you picked anything else, that would be wrong. I could ask you, you're looking for amylose in a frog. Your friend says, I don't think you'll find that there. Is he right? Will you find amylose in a frog? Why not? It doesn't store its energy as amylose. What would you find in a frog? As a starch, as a glycogen. Good. All right. Cellulose. So this is found in plants, and this one is built with beta glucoses. Does that make sense? So this is starch, which equals carbs, which equals amylose slash pectin. And also this is glycogen. This is found in animals. This is found in plants. This is cellulose. Where's that found? Plants or animals? So cellulose is made up of beta glucoses. So what would we call those kind of bonds? Do you think? If these are alpha bonds, what would that be called? Beta bonds. Good. So cellulose is just like starch in the fact that it's all alpha 1,4s, but it's not made out of alpha, right? So it's beta 1,4s. And what is this called if it's on the same side? And this is good. I promise you I'm going to put a molecule like this and I'm going to ask you, is that cis or trans? So make sure you guys know that. CIS. C is in cat. I is in iguana. S is in snake. Those are the ones I use because I'm a biologist. Not Charlie. Alpha. It's alligator. So these have to alternate to make this structure. So this one's up, this one's down, this one's up. This one would be Sesame Street pattern thing. So if we look at starch, these are all down, and that's what makes it turn. So this one looks a lot different, and it behaves a lot differently. instead of curving, this makes flat structures. And in doing that, it allows these stacks to form hydrogen bonds. So next time you guys are in the lab, take a look at a piece of notebook paper. You'll see that it looks just like this under the microscope. And it's made up of all of these fibers of these, what kind of glucose? Alpha or beta? Beta, what kind of linkages? Beta 1,4s. So these beta 1,4s are laid out flat, and the hydrogen bonds allow them to be strong, right? So what do you think stronger, glucose or cellulose? Go run into a bag of sugar and then go run into a tree. Which one's stronger? So this is what allows trees to have that really strong external cell wall. That's what gives them rigidity and strength. That allows them to be out in the elements and the wind and stuff without falling over. Because if you're a tree and you fall down, it's not good. So these are the hydrogen bonds. Remember, oxygen and hydrogen, they don't share well. So where does the electron spend the most time? Around oxygen, and that makes that partial charge, right? So oxygens are attracted to hydrogens even if they're not in the same molecule because they have a partial charge. Hydrogen's partial charge is positive, oxygen's is negative, right? And opposites attract. So beta 1,6 is not, we haven't even talked about that. There's not in anything that we're going to cover.
Except that's where nitrogen comes in.
Right, but that's alpha 1,6. So that's between the number 1 carbon and the number 6 carbon. No, they're all 6 carbons. So
It's just where the bonds are.
yeah, let me just go. I'll go back so I can just talk about that. So here's the numbering system. So this is 1, 2, 3, 4, 5, and that's the 6 carbon. Okay. So, and again, 1, 2, 3, 4, 5, 6. So this one is alpha 1,4 because it's from. You can see that these are cis, right? And that's, I can tell you right now that that's alpha glucose because it's cis. Okay. So this is automatically an alpha bond because it's between alpha glucose. And then the number is the bond between number 1 and number 4. So a 1 and 6 would be between this and that. Does that make sense? You know, or a 1, 3, or whatever. This is really important because when we're synthesizing DNA, and this is how your DNA is sequenced, right? This is how we copy DNA. The numbering system on this sugar is called ribose. Ribose has, there's a carbon here, so it has, remember we number clockwise? So this is number 1. The number 1 carbon is where the base is found. So this is the A's and the G's and the C's and the T's. this stuff is made of parts, right? Your DNA is made of parts, A's and G's and C's and T's. So let's say this is an A, and I want to add a C on the end of that. Where do I add it? Where can I add it? First, think about the reaction. What's the reaction we have? Okay, so if we're going to take out water, what do we need? Tell me what water's chemical formula is. H2O. Can you make H2O from this? Can you add DNA to that end? No. Can you make H2O with that? So you can only add DNA to the 3' end. And that's what we're doing in lab, because DNA can only be copied in that direction. Yes. And so, this is how they find out. They copy the DNA, right? Because they want to find out, if OJ left his blood all over the place, or, what. I mean, I'm dating myself because that was like when I was in grad school. But, you get my meaning, right? Or who the baby's daddy is. All this stuff wouldn't be possible if scientists didn't think about this sort of thing, like what direction DNA is going to be copied. Because, if I sent you down the road, I just spun you around and said, I'm just going to send you in one direction, and I want to send you to Flagstaff. You'd want me to go in the right direction, right? You wouldn't want to start copying, thinking you're going to go this way, and really end up going that way. That make sense? Hopefully it will later. We're going to do a lab on this. But that's why the numbering system is important. It's used all the time, and it makes a major difference. Yeah, so that makes it branched, yeah. And you could build these molecules at home. You could get, grapes and toothpicks and stick them together if you want, and you'll see that's how they behave. That's what they look like. That's why I wanted to do the chemistry lab later instead of the second week, because you're like, what the hell? I'm just putting sticks and balls together. But this is important stuff. That's why it takes up half of your book is the chemistry behind all of it. All right, so cellulose. So you guys get the gist for the test. I'm going to ask you, what kind of bonds are found in cellulose? What are you going to tell me?
Beta 1.
What kind of bonds are found in glucose? There's not a glucose. Glucose is by itself, so those are just covalent bonds. What kind of bonds are found in amylose? Now you can tell me what you're going to tell me. Alpha 1, 4, right? And amylose. What about amylopectin? Alpha 1, 4 branch. What about glycogen? All right. And the last one is chitin. So this is what makes up, the hard shells of insects and crustaceans like lobsters and stuff. Can you eat lobster shells? Can you live off that? So you can't digest lobster shells. You can't digest cellulose. This is made out of beta glucose 2. You guys get the pattern? That's right, your enzymes don't recognize the beta bond. They don't fit on there, so they can't break it down. So you can't use any kind of beta bonds as an energy source. You don't have the enzymes for that. If you were a termite, technically a bacteria living in a termite, then you would have the enzymes that would break that down. But animals don't have that. Yes? So that aspirin is, yes, it's a chemical that's synthesized from that. In fact, so is the breast cancer drug Taxol. It's made from the bark of a tree as well. But it's not cellulose. It's something else. All right, so this is different. I don't care that you know this. This is an amino group. You need to know that functional group, right? So on the test, I might write this NH2 and ask you what group is that? What functional group is that? NH2. It's an amino group, right? So this has a modified amino group linked to it. It's actually called an amide, but don't worry about that. Just know that it's beta 1,4. So on the test, I might ask you what kind of bond is found in chitin. You guys get the idea? All right. So any questions about this stuff? That's why it's important that you guys know the stuff that we learned before, like geometric isomers, because you don't know what the heck I'm talking about if you don't know that that's a geometric isomer of that. And then you can't tell me what the difference between cellulose and carbohydrates are. What's the difference between fiber and carbs? One you can use for energy, the other one you can't. Which one? Alpha or beta? Which one can you use for energy? Alpha, good. Alright. So, lipids. You guys know what lipids are? They're fats, right? But there's different kinds of fats. Why? What makes fats unique? They're not water soluble. Why aren't they water soluble? Kind of. Be more specific, please.
Yeah.
So remember back in chapter 2, we were talking about different kinds of bonds. What's a bond between two atoms that are sharing electrons? What's that called? Covalent. And there's two different kinds of those bonds. Polar, non-polar, right? So we talked about like dissolves like. What's water? What kind of bonds are those? Polar or non-polar? And what's the bonds between, well, fats are made of carbon and hydrogen. What kind of bonds is that? Are you sure? Alright, so for the test, I'm trying to prep you guys for the test so you kind of see where your strengths and weaknesses are. So on the test, remember I told you, I'm going to tell you the electronegativities and you need to be able to tell me what kind of bond it is. This is 3.5, this is 2.1. What kind of bond is that? You're right, it's polar. This one is 2.5, this one is 2.1. What kind of bond is that? How do you know? It's smaller than 0.5, that's right. So this shares equally, so there's no charge, right? These share unequally, so this gets a slight negative charge, and this gets a slight positive charge. The reason water can dissolve things is because it can surround it. And the only things it can surround are things that have a charge. That's right. So, does that have a charge? You just told me it's non-polar, so you should know it doesn't have a charge. So if it doesn't have a charge, can water surround it? Then it's not water soluble. Is DNA water soluble? Why? Because it has a phosphate group on it, and that therefore it's negatively charged. So water can surround it because water can surround anything that has a charge on it. Good. So what these all have in common is they're not water soluble because they're made of carbons and hydrogens. They don't have a charge, they're all nonpolar, and they can't be dissolved. So there's three different groups: fats, phospholipids, and steroids. So we'll start with fats. Fats are, what you store in your body, right? As fat, which you use as an energy reserve. So in the scientific term, those are called triglycerides, or the technical term is triacylglycerol. because it's three. The tri means three, right? Everybody knows tricycle has how many wheels? So that's acylglycerol, which the acyl refers to the fatty acids, which you guys built. You probably don't remember this, but you built these in the lab. Except you built little short fatty acids, like 2 carbons long. They don't normally exist that way. They're normally like 16 to 18 carbons. So this is glycerol. If this didn't have OHs on it, it just had H's on it, what would that be? That's right. Methane, ethane, propane, 3 carbons. So this is glycerol. It has that name because it has OHs. Remember, adding an OH makes it, what's the term for the functional group? An alcohol. And alcohols end in OL. So glycerol is the name for that. It's propane with 3 OHs on it. Remember the name of that functional group? Hope you know it for the test. Hydroxyl. Good. When we want to add, when you guys did this in lab, you want to add fatty acid to a glycerol, what's the reaction? You want to build molecules, what's the reaction called? Dehydration. And if you want to break it down, so if your body's building fats, it's going to do dehydration reactions. If it's breaking them down to use them for energy, what's it called? Hydrolysis. Good. Alright, so this is it. Triglyceride. If you went to the doctor and got your triglyceride level test, that's what they're looking for. They're looking for the amount of fat in your blood. And this is what they're looking for. So 3 fatty acids linked to a glycerol head, and that is called triacylglycerol. And there's different kinds of fats, right? You guys know that? So sometimes you call them oils or whatever, but you go down to the grocery store aisle, right? There's a whole aisle with different kinds of oils. There's olive oil, there's canola oil, there's rapeseed oil, linoleic oil. You go further down, what else is on the aisle? What other kind of fats are on the aisle that you could buy in the store? Okay. And then there's animal fats, right? Like lard and butter and stuff like that. So all of these are fats. They're all triglycerides. What makes them different is the length and shape of their the fatty acids in them. That's the only thing that makes them different. Right? And you probably heard all these fancy terms: saturated, unsaturated, mono, unsaturated, trans fat. What's that? What's hydrogenated mean? What does all that stuff mean? You heard it all, haven't you? What does it mean? You just like, what's the good one and what's the bad one? Do you know? It has to do with the double bonds. You're exactly right. So this is what makes fats different, right? So if you're talking about a saturated fat, this is an example of a saturated fat. So saturated simply means that it has the maximum number of hydrogen bonds that it can load on there. So if you have 2 carbons, a fatty acid chain with 2 carbons, doesn't really exist, but let's just pretend. I put as many hydrogens on there as I can. How many hydrogens can I fit on there? 2 carbons linked together. Alright. You guys don't want to play? Fine, I'm throwing it. 2 carbons. How many hydrogens can I load on that thing? Why can't I put more than that? Right, what's the maximum number of bonds carbon can make? 4. So that's about 4, that's got 4. I can't legally add any more hydrogens to that. It won't. It's too many bonds. Alright, it's not permissible in chemistry. That's saturated, right? So, but I can add a double bond into that, and then that's unsaturated. So saturated just refers to the term as it has as many hydrogens as it can hold. Now, if I took this fat and I hydrogenated it, what would I be doing? Adding hydrogens, right? So when companies hydrogenate oils, they're turning them into saturated fats. Does that make sense? All right. I'll explain to you why they do that in a second. But everybody understand the difference between saturated and unsaturated? All right. Okay. So saturated fats don't have any double bonds. So they essentially lay flat. Their shape is like so. If I add a double bond into a fatty acid, it causes it to bend. Right? So if I was putting papers on top of each other, let's say I had 500 sheets of paper and I stacked bent ones on top of each other, randomly bent, not perfectly bent, but, and then flat ones, which one would take up the most space? The bent ones, right? Everyone agree with that? So if something takes up more space with the same amount of mass, you say that that is less dense, right? And if I took 500 sheets of crumpled up paper and threw it at you, you would probably appreciate that more than if I took a big ream of paper and chunked it at your head, right? Why? Because the ones in the flat pieces of paper are more dense, right? It's just like a snowball fight. You wouldn't have a snowball fight just off the ground and throw it, would you? What do? You pack it together. This is the same way it works in the molecular level. If I have a bunch of 3 fatty acids stuck together versus ones that are bent, which ones are going to be more dense? The flat ones, right? So they're more solid at room temperature because they're more dense. Right? So when I put a double bond in there, they bend, they take up more space, that makes them more liquid at room temperature. So which one do you think are solid at room temperature, saturated or unsaturated? Saturated, yeah. That's what makes up animal fats, right? Butter, stuff like that. And then if they're liquid at room temperature, what do you think those are called? They're unsaturated because we have a double bond and it's bent. All right, so plant fats are easy to get at for companies than animal fats. Everyone agree? But they have different consistencies, right? So if you want to make your ice cream more delicious, you use more fat, right? More cream. But if you wanted to, you didn't want to have to go and get animal fat, what could you do to turn plant fat, which is the unsaturated, into animal fat, more consistent animal fat? What would you do to it? This is liquid at room temperature. You want to make it more solid so your ice cream is more creamy. What are you going to do? You want to add hydrogens, right? And so that's hydrogenated. Make sense? So saturated fats, no double bonds, solid at room temperature, animal fat, straight chains. Unsaturated fats, they're the same, except they have double bonds that cause them to bend. So there's monounsaturated fats. What do you think that means? I'll give you a hint. It has to do with the number of double bonds in it. Yeah. And then usually you don't use di, but poly, 2 or more. So that's the difference between mono unsaturated and poly unsaturated. And the more double bonds you have, the more bent it is, the more liquid it is at room temperature. So the thinner it is. That make sense? The more it bends, the more space it takes up. So now let's talk about trans fats. You guys heard of those? Are those good for you? Do you want those? You want a big plate of trans fats? Why not? What's wrong with them? You guys just watch TV and you're like, oh yeah, those trans fats, they're bad. Well, I don't even know what the hell that is. Alright, so this is one of the reasons that you guys need to know that cis and trans term, right? So we have this long chain of carbons, and all of a sudden we have a double bond, so it can go either way, right? It could bend this way, or go that way. Everyone agree? And then I have 3 bonds in this carbon. That's not allowed. What do I have to do? No. Add hydrogen. This one, 3 bonds, not allowed. This one, same thing. What kind of fat is that? What kind of fat is that? Ooh, why is that bad? Look at the shape. What takes up more space? Which ones can you stack on top of each other more easily? So that makes them more solid at room temperature, so they're... What would you rather have flowing through your blood? Stuff that's solid at room temperature or stuff that's liquid at room temperature? That's why trans fats are bad. These are made, this isn't naturally occurring. This is made through the hydrogenation process because manufacturers want this kind of fat to look like animal fat. That's all they're doing. It's just a simple trick. Chemistry. But it's not good for you because it's solid at room temperature. It simulates animal fat. That's why trans fats are bad. Now, see, you learned something.
Devastating numbers coming from those.
Sorry to hear that. Just might be honest. All right, so fats have a purpose, right? We need them to store as an energy source. They provide cushion for our organs, right? So if you tripped and fell on your kidneys, you wouldn't pee blood for a month because your organs are cushioned by the fat. And then they insulate you against, temperature loss, so they help you maintain your heat. Like you guys know that from seals and whales and things that live in the Arctic. All right, any questions about those? All right, so the next one we're going to talk about are phospholipids. This is very similar to what we just talked about, except this has how many tails? Fatty acid tails? Count them with me. 1, 2, And triglycerides have how many? 3. Sickle. How many wheels? Dang it. Is that why your hand's up? This one's not going to come on because the light's still flashing. So I have to direct your attention to the screen. Sorry about that. Okay. Where was I at? Okay, yeah. So, triglyceride, 3 fatty acid tails. Phospholipid has 2 fatty acid tails and a glycerol head, right? And so, we talked about this, and I want you guys to understand this. You could just memorize it, but that's okay. But this is hydrophilic. What does that mean? And that means it can dissolve in water. Now, why can that magical chemical there dissolve in water? It has a charge. There's a charge. There's a charge. Has a charge. Water can surround it, right? If it doesn't have a charge, then this one is hydrophobic because right, but the reason that it doesn't dissolve in water because it doesn't have a charge, because it's a covalent non-polar bond. Okay, so this sort of molecule has a dual nature. It has hydrophobic tails and a hydrophilic head. If I were to take this and put it in a bunch of this in water and shake it up, what kind of orientation do you think it would take on? Okay, I got a bunch of these in water. How are they going to orient themselves? So all the tails are going to be afraid of the water, right? So they're going to hide, and all the heads like the water, so they're going to be on the outside. So these things self-arrange into these things called micelles, which looks like this, right? So, now we're thinking about, like, where did the first cell come from? Because this is actually the components of cell walls. Cell membrane. Cell walls are made out of what? Cellulose. So this is made up of phospholipids, and you want these to be made out of fats, right? If they were made out of water, what would happen to the cell wall? It would break down into the cell and collapse. Could you maintain homeostasis? Because that's the reason you have a cell wall. No. It'd be like this trying to make this room a different temperature than the outside with no walls around it. So we need this to be made out of fats, right? But it has to be a special kind of fat because you need to have water inside the cell and outside the cell so you can move stuff around, right? Because there's no like little guys in there that are moving, your sugars over here or anything like that. Water is what causes it to move around in there. And that's why we need water to live. We already talked about that. So I can't make it out of one of these layers, but I can make it out of two. If I have the heads facing the water on the inside of the cell and also facing the water on the outside of the cell, this inside is going to be philic or phobic? Phobic. And now we have a barrier, right? That works really well. We have water on the inside and water on the outside. So this is how your cell walls are made. They're made out of phospholipids. And is it a single layer? It's a double layer, which in Latin or Greek we use the term for two as dior. I'll give you a hint. It has two wheels. Bicycle, right? So so this is called a phospholipid. Bilayer. That's what cell membranes are made of, which is shown right here. And this sets up another problem, right? Because I can spin this one. Because now you need to get water inside your cell, right? But you just set up a whole membrane that doesn't allow water to go through it. In fact, nothing can get through that. So how do you get stuff inside your cell? If I built walls all in this room, how would we get in and out? You need a hole, right? A door or window to climb out. So your cell makes little channels in it, right? And those channels only allow certain things in. Kind of like nobody could get in except public safety push some magic button and change their light from red to green so we could get in the room tonight. But that's how cells work, right? They're not always open to the outside. So let's say, you guys probably know this because if you eat a bunch of glucose, what happens to that? It just magically gives you energy. So why do you put it in your mouth? Because you have starch, right? You put it in your mouth, you break it down, turns into glucose, goes into your blood, and then what does it do once it's in your blood? So you could ask a diabetic, it stimulates insulin, right? Insulin stimulates a channel to be made that allows glucose to come in. So glucose can't come in unless insulin is present. So that channel comes and goes based on the presence of insulin. That's how your body can select what it wants to come in and out of that cell. We call that selectively permeable membrane, and we're going to cover that in chapter 7. Any questions about that? Okay, so the last fat that we're going to talk about is steroids. There's two different kinds of steroids. There's what we call lipid steroids, and then there's protein steroids. So insulin is considered a steroid too, but it's not a lipid steroid because it's made out of protein. This is considered a lipid because it's carbons and hydrogens, right? So is it water soluble? Carbons and hydrogens, are they water soluble? No, they're not. So that's why it's a fat, right? That's why it's in this category. So all steroids are synthesized from cholesterol. We looked at 2 steroids already, right? In the last chapter, remember? Testosterone that makes you male, and estradiol or estrogen that makes you female. Those are all synthesized from cholesterol. So what happens to bodybuilders and, especially female bodybuilders when they drop down to like a really low percentage of body fat? Anybody know? Right. Well, you can't produce steroids. So, like for females, if they lose a certain amount of body fat, they don't have a menstrual cycle anymore because they can't produce the steroids that are required to do that. So you need cholesterol, right? I got on my big soapbox about, this is a big myth or whatever, but I want you to think about that. So we're talking about this molecule here, which is a fat. But if we're talking about LDL and HDL, what is the term that's used for that? What does HDL and LDL stand for? Do you know? High density lipoprotein. What is that? Is that a fat? What are proteins made out of? Amino acids. So HDL isn't even cholesterol. It's the molecule that has to carry cholesterol in your blood. Why does it have to do that? Because cholesterol is not water soluble. Does that make sense? So cholesterol is the basis for all the steroid hormones that are synthesized. So here's some example. Here's cholesterol. This is another hormone called 7-ketocholesterool. Here's testosterone. Here's estrogen. Notice they all have the same ring structure. Right? And I'll just close real quick and I'll show you guys that. Those aren't the only ones. Oh, we're way over, huh? Alright, well, I'll just stop here and I'll show you the thing later. But all steroid hormones are synthesized from cholesterol. So cholesterol is really actually a very important molecule. You couldn't live without it. Alright, so the plan is we're going to finish up chapter 5 on Thursday, and then we'll have a review on Tuesday, and then you guys will have your test on Thursday. I'm a little worried about you guys. You might want to think about starting studying a little, just a little.